Okay, fantastic. Yeah, fantastic. So I slightly changed the title of my talk. It's basically the same as sensing and computing with liquid marble, slime mold, and fungi. But I call like main title Neuroscience without neurons because that's what I'm most interested about right now. And what I'll be talking about spiking in systems, which actually not neurons at all. They are the chemical systems or plants or fungi or slime molds, and how we can do sensing and computing with the spikes. And on the first slide, you can see basically action potential, which is common for neurons. We have depolarization, repolarization, and refractory period. And it's about like 5 milliseconds, the whole spikes. As we will see further, the same spikes, which are before thought only characteristic of neurons, can be observed in liquid marbles with Belov-Zhabotinsky medium, in fungi, in plants, and in slime mold. So we will talk about liquid marbles with BZ medium, slime mold, Physarum polycephalum, fungi, mostly oyster mushrooms, and the plants. With regard to plants, it will be a model of the computing on the vascular system of the single leaf. Okay, chemical oscillators. We all know, well, majority of those who kind of into complexity science and nonlinear systems know Belov-Zhabotinsky medium. It's organic oscillatory chemical reaction where in 2D and 3D we can observe waves of the oxidation of the medium. They're seen as blue in the picture. And if we insert electrodes in the medium, we will see spikes. Just a moment, I will show this picture and we return back. Yes, this picture. So you can see propagation of oxidation waves and electrodes inserted somewhere on the top, not visible. And basically because electrode is quasi one-dimensional, we can see spikes similar to action potential. So that's more kind of more videos of this BZ marble, BZ liquid marble. It's basically done by taking a soft solution of a BZ medium, like droplet 50 milliliters, and rolling it into hydrophobic powder. And then hydrophobic powder covers the whole kind of liquid droplet. And then we get liquid marble, which is basically the soft body and can be manipulated by your fingers and rolled on the surface. And you see propagating oxidation waves in this marble. And in our experiments, we measured basically oxidation waves as spikes by inserting 2 electrodes in the liquid marble. And you can see on the picture liquid marble lying and 2 electrodes are inserted. So oscillator is basically one of key components of the electronic circuits. That's why it's important to study oscillation in liquid marbles with that medium. To use oscillators, we must somehow control frequency of oscillation. How this can be done? That's one of the examples of how it can be done. It can be done with freezing. So we put Peltier element underneath liquid marble with that medium, and then we measure change temperature and measure oscillations. What we find is that when we cool down liquid marble, the frequency of oscillation decreasing. When we warm it up, frequency increasing back. Just by changing temperature, oscillations can be controlled. Also, this can be done with light, because light basically is inhibiting component of the reaction. Therefore, if we eliminate it, then oxidation waves will stop and the frequency of oscillation will decrease. On these 2 plots, we can see on and off. Light is switched on, oscillation stops, and light is switched off again, and oscillation continue. And on the below plot, we can see repeated switching on and off of the light and ceasing and restoration of the oscillations. Thus, we can see just more kind of advanced experimental plot, and right vertical lines, it's part when light switch is switched on. And also when light is switched on, the actual shape of the spikes also changing. And if we can detect shape of the spikes from the electrons, then we can know without actually looking at the light that light is switched on. So this phenomenon was used into control of the robot. So what we have done, we placed liquid marble on top of the robot. You can see on the right picture. Insert 2 electrodes and made Arduino board, which actually reads the spikes and detect spike shapes and frequency. And then the subwheeling board send command to motors. There are two motors of the robot, and which move robot forward and rotate the robot. So that's one video, how it works in reality. So robot moves, and when it will be illuminated, you will see like green laser light shining on the robot. Hopefully there will be soon some of the pictures. Okay, you see green light is illuminated, and then as a response to illumination, oscillations changed in this busy droplet, and robot also rotates. That's how basic robot can be controlled. Okay. That was to start with. Right now, we are still continuing experiments on trying to couple oscillation with marbles. As soon as we'll be able to couple oscillations, where frequency of oscillations on a marble change frequency or affect somehow of oscillation in another marble, then we'll be able to do a pattern recognition on the array of BZ marbles. But that's kind of subject of the future studies. Now we switch to slime mold briefly. Well, slime mold is single cell, like Fusarium polycephalum, for example, which propagates in the space, as you can see on this photo, occupies source of nutrients and connects them with protoplasmic tubes. Slime mold is mostly known for solving special problems, for example, like maze solving, like in this video. It finds paths inside the maze to the source of attractants. And despite the fact that basically it does find some paths, but it's not the shortest path, nor the longest path. It's some kind of average path. Anyway, in this particular talk, we will not talk about solving of spatial problems, but focus on the oscillations of electrical potential in slime mold. So, slime mold can spike in a similar manner as neurons can do. If we grow slime mold between two electrodes and start recording voltage of extracellular potential difference, indeed, then we will see there are spikes, as you can see on the right plot. Amplitude of the spikes depends on the slime mold mass and how good its connection. So it can be like amplitude 8 millivolts or 10 millivolts, even 20, and the period between 60 and 120 seconds. Also, spikes can be caused by mechanical stimulation, by light, and by electrical stimulation. What most cool is that slime mold somewhat respond to electrical stimulation in a manner similar to neurons, as you can see on the spot. We have oscillations of the slime mold before stimulation, the stimulation with periodic function, and then a response to stimulation with increased amplitude and decreased frequency of spike. We indeed can couple blobs of the slime mold or blobs of the agar gel occupied by slime mold together electrically, and then we will experience more complicated patterns of the oscillations. That's the exciting field that could be explored further. At the moment, we just tried with 3 blobs and then switch to other project, but I believe in principle we can make a simple neural system made just exclusively from the blobs of slime mold. What most cool, in theory, when we do experiments with amplifiers, because voltage is very low, like 5 millivolts or 7 millivolts. However, if you just apply direct voltage to the slime mold, then output potential will be amplified, and then we don't need any particular amplifiers. For example, when we apply like 15 volts, as on plot D on the picture, we have about 10 volts response, which is basically enough to measure with any device we have even at home, just handheld multimeter. That makes electronic circuits made from slime mold very handy and very kind of easy for mass production, at least for entertainment. Right, what next? Tactile sensor. That's a very nice example of what can be done with slime mold. Like spiders, they have tactile sensors. And they make like growing part of cuticula, and inside cuticula we have dendrite. And when this hair made from cuticula is moved, then due to like twisting of membrane, we have action potential emerged, and then neuron fires and transfers information to other neurons. Can this be done with slime mold? Yes, it can. If we take 2 blobs of agar, put into electrodes, insert hair, just normal human hair, in one of the agar blobs, then slime mold will occupy, as you can see on this plot, these hairs. Okay, so we already have imitation of the spider hair, but slime mold is like dendrite, but not inside the hair, but outside. And when we touch the hair, like we see on the example of the rat, we made this like hairs, and then we kind of touch them, and that was response. When hair is touched, we have very strong action potential, followed by a wave packet of small oscillators. And it's like persistent. Therefore, tactile sensor can be made from slime mold just based on the action potential and oscillations. Reservoir computing. In the reservoir computing, we can apply binary strings represented either in high and low voltage or low and high frequency of periodic function, relative periodic function. And then we measure response of the slime mold. And then again, low and high voltage can be interpreted as bit up or bit down. So in fact, we can map sets of binary strings onto slime mold represented by electrical parameters of the stimuli and read back binary strings represented by electrical properties of the slime mold, which emerged in response to stimulation. So we did this experiment with slime mold. That's distribution of the logical gates discovered. Indeed, as always, a XOR gate is a most rarest gate, not just with slime mold, but also, as we found later, with plants and fungi and chemical medium. And other gates are kind of more common. Right. So what with slime mold, we kind of logically start from sensing, and then with reservoir computing and how to implement a Boolean circuit. Many inputs, many outputs. What can be done with plants? Experimentally wise, not so much. However, in theory, we can already demonstrate that computation can be implemented on a single leaf of the plant. This can be done as the following. If we insert electrodes in the vascular system of the plant, we observe action potential like impulses. These impulses can be imitated with Fisk-Hughes-Nagumo equations. That you can see example of how excitation wave imitated with Fisk-Hughes-Nagumo equation propagates in 2D. There is a so-called excitability parameter in the Fisk-Hughes-Nagumo equation. By changing this parameter, we can switch excitation from normal circular waves, as you can see on the plot A, for example, to quasi-solitons. And these quasi-solitons can propagate for the substantially long distances. Indeed, as with those solitons, they will not survive forever. So either they explode and we have like circular wave, or they just die out. Anyway, if we select the right parameter of excitability, we can propagate excitation in the direct manner for some substantial distance, substantial enough to collide it with another wave and implement computation. So that's propagation, another example of propagation. And now we go to the leaf. That's vascular system of the real leaf. And we assume that conductivity of the passes depends on the width, basically. And we insert imaginary electrode or simulated electrodes in some part of the vascular system. And then we stimulate the leaf in some of the parts to imitate basically inputs in our logical circuit. And we record spikes. You see spikes recorded from different part of the system. Then, depending on the coincidence with the spikes, in response to binary inputs, we can encode each of the gates, each of the electrodes as a logical gate, as we can see in this example. And then we can try basically to implement logical function and advance further experimental circuits implemented in plants. That's just photos of some experiments which are ongoing right now. Unfortunately, with plants so far, we observe substantial amount of noise. Therefore, further work is required to actually filter the noise and select spikes and non-spikes from the system. Talking about plants, at some point we tried to make actually brain from plants in a quite bizarre manner. We made brain from agar gel and then put seeds of the lettuce on this agar gel, allow them to grow, and hope that at some point this seedlings will start to interact with each other, which will be reflecting the electrical activity. So we recorded indeed external kind of electrical potential from this lithius brain, and we found that there are some kind of waves of electrical activity somewhat similar to electroencephalogram of the real brain, and at least there is a reaction, electrical reaction to stimulation with light and with tactile stimulation. You can see on this plot its response to the light on and light off with different spikes of electrical activity. Okay, now we're switching to fungal sensors and fungal computers. So how I started, at some point a couple of years ago, I thought, all kind of living systems, they exhibit electrical activity. What will happen if we just insert electrons in floating bodies, particularly mushrooms or fungi, and record for some time. So I have done it with different types of fungi, and I found that if we insert electrode in the one electrode in the cup, another reference electrode in the stalk, as you can see on these plots, then we observe action potential. This action potential is remarkably similar to that observed in neurons. We have depolarization, repolarization, refractive period, as you can see on the picture. However, comparing to neurons, that's another measurement. You can see in the growing box, and there is a container with substrate inoculated with fungi, and we just insert electrodes in the substrate. Yes. However, width of the spikes are much wider. If you remember, action potential of the neurons will be about 5 milliseconds, right? In our case, it can be anything between 5 minutes and 20 minutes, just a single spike of the fungi. That's because fungi is essentially immortal. We know, like, largest mycelium is about like 100 hectares in Oregon of one species of fungi, and by genetic analysis, it's about 200 million years old. That's how old is fungi. And when you live 200 million years, you don't need to rush because you can live forever. Therefore, mushrooms think very, very slowly, much more slower than us do, because they have no one to rush. Yes, and this you see example of the spiking, and basically we have like spikes, and they can be grouped in trains in the same manner as trains of spikes in real neurons. That's examples of trains of spikes. And as you can see on this plot, there are like high frequency oscillations and periods of 125 minutes, 2 hours. And on the right kind of plot, zoom, we have relatively low frequency oscillations, and distance between spikes is about, well, 300 minutes, well, a bit more, so like 5 hours. That's how slow thinking fungi actually are. So we also, Muhammad Dekhshebi, my mate and collaborator, analyzed complexity of the spike of fungi. We had about like 600 records, experiments, and each experiment we recorded about 8 differential channels of the electrical activity of the fungi. He wrote a customized program to recognize spikes in fungi, because normal programs written for neuroscience, they don't work, because spiking activity of fungi is very, very variable. So yes, he detected spikes. Then he found like that more or less width of the spike doesn't depend on the width between electrons in the differential pair of electrons. And then he decoded the spikes into binary representations and calculated many measures of complexity, including indeed Lempel-Ziv complexity based on compressibility. What was found, a part of like many interesting things which you can read in the paper, it was found actually that fungal language expressed in the binary strings derived from spiking is very complex. You can see here, for example, complexity, algebraic complexity of European languages. And you can see that the most complicated language will be Finnish language on the right. And if we put LZ complexity of the mushrooms, they're even higher. So based on this one, we can speculate that basically language of mushroom is more complicated than human language, at least if we consider European languages. What else mushrooms can do? There is also such thing as fungal skin. We can grow mycelium on the liquid culture, and then we'll have like a sheet, two-dimensional sheet of the mycelium, just pure mycelium. And it's about 2 millimeters thick, can be 4, and it has structure of the skin and feels like a skin. And it can, in normal condition, can stay alive for a very long period. So right now, our colleagues in other countries are different experiments on making like wearables from the mushroom skin and making mushroom skin for robots. On our parts, we decided to check sensitive properties of the skin. Therefore, we insert electrodes in the skin, you can see on this picture, and applied weights and light to the skin. You see in this plot, W with star, it's when weight was applied, we put some object on the skin, and WO, weight is removed. The same with L star, light is switched on, and then LO, light is removed. Even on this plot, you can see that one object is placed, then fungi react with the spike of electrical potential, followed by some kind of train of smaller spikes. And then again, normal activity. When weight is removed, we have the same reaction, like switch on-off. If light is applied, then overall action potential, electrical potential goes up, then stays at this level, then goes down. Because that means that actually fungal skin can differentiate between types of the stimuli. Right. What next interesting we have done? Indeed, fungal skin can respond to stretching. I think I mixed our slide. Yes. We did experiments with mannequin. We inoculated hemp pad and attached this hemp pad to the normal shirt. And then we just applied some weight to the shirt to check what will respond to stretching. And fungal skin respond to chemical stimulation like malt extract with increased frequency, respond to stretching with increased frequency, but of different temporal measures, and respond to stimulation with ethanol, for example, with very strong action potential, followed by trains of spikes. And again, in this case, differentiate between stimuli. Okay, more experiments on fungal response to loading. Yes. So we had this block in our experiments. It's a block of substrate, like hemp substrate, colonized by a Ganoderma lucidum mushroom. And we insert electrodes in the sides, record at normal activity, and then put 16 kilo kettlebell on the top of this block and record activity again. And then remove kettlebell, record and put back, basically. We tried all possible combinations. So what we found? We found that you see this on and off, it's moment on the plot when we placed weight 16 kilo and removed weight 16 kilo. Response to the application of the weight in spike, followed by train of spikes and change of the frequency of oscillations. So you see on and off spike amplitude and duration, how it's changing during stimulation, the same for off spikes amplitude and duration. What's most interesting, it's not like fungal substance can react to the weight. It's like we can observe habituation. So when we repeat stimulation for many times, then response in terms of amplitude and medium duration of the spike goes changing. So basically, similar to neural systems, our fungal brains, they gradually adapt to stimulation. Okay, now we can also do computation with spiking fungi. Experiments in the lab are continuing. What we have done so far, we have done simulation. Our colleagues extracted three-dimensional single fungal colony, as you can see in those photographs, very small, about 200 micrometers. Then we inserted simulated electrodes, array of electrodes, as you can see on the right plot, in this fungal colony. And we simulated propagation of the excitation, calcium waves more likely, using Fiske-Lagom equation. Here you can see plot of the excitation propagating from one single point on this colony. Indeed, when excitation propagates on the colony, electrodes detect spikes. We can take two electrodes, assume the impulse electrodes, convert 0 and 1 to no impulse or impulse, and stimulate the system. Then, depending on the coincidence of spikes, as you can see on this plot, we can interpret the spikes as logical gates. So in each electrode inserted in the colony, one type of the logical gate is implemented. Moreover, during development of the system, when excitation continue propagating of the system, more and more gates appear. So gates not just fixed, they kind of dynamically changing. And trying to click back to show you table. Yes, everything is so suddenly slow. Yes. So we calculate frequencies of the gates. Again, XOR gate is like most rare gate. But what we found? We found that for most living systems, distribution of the types of gates computed is almost the same. You can see on this part, the gates computed with Physarum polycephalum on succulent plant, cacti, on single molecule of protein vertaxin, done in our other computation experiments, and on actin bundle networks. And they more or less are the same. So it means all living systems, they have similar nonlinear properties, which are essential for site distribution of the logical gates. Right. And now final kind of remarks about spiking cytoskeleton. So we all know that neuron spikes, but essentially cytoskeleton, actin filaments, and tubulin microtubules, they also spike. And here, example, we took tubulin microtubules, placed them in a small droplet and insert electrodes. And we recorded potential difference between two electrodes. Distance between electrodes was about 2 millimeters, and we observed very, very nice spiking, as you can see on the zoom on this plate. So, as you saw in the previous demonstration of computing with single fungal colony, the same approach can be used to compute with bundles of actin filaments. So on this picture, you can see bundles of actin filaments, just one of the slices. Essentially, it's like 3D structure. And if you insert electrodes and simulate excitations of actin filaments with Swiss Naguma, we can see, for example, the following development. We excited in one point, then we have propagation of the excitation waves, and those with numbers are electrodes, many electrodes, and these propagating waves on electrodes, they reflect the spikes. So just show next picture, probably will be a slide with the spikes. Not yet, okay. And then again, based on the coincidence of spikes derived from binary strings represented by two input electrodes, we can interpret the spiking as gates, as you can see on this table. So you see a black spike which responds to input 0, 1, red one 1, 0, and then green 1, 1. Right, and that's what you can see basically when you like unroll temporally what was recorded on the electrodes. You can see this like waves propagating. They are represented by spikes. And in terms on each electrode, at each period of time, based on the occurrence of spikes, we have one of the Boolean gates or functions implemented. That's example, basically what you see on the one, just one select electrode, with electrode number 7. And then we can like check all gates implemented on the electrodes and look at the frequencies of the spikes. Just example table for one of the experimental setups. Again, gate source is quite rare, other gates are more common. But anyway, it's doing it. And returning back to the title of the talk, brains, neuroscience without neurons, or fungal brains, we ask ourselves, are mushrooms consciousness? And in 2018, when I was in the conference on consciousness in Lausanne, we discussed with Stuart Kamerov what is consciousness, and eventually it was agreed that if a thing can be anesthetized, the thing has consciousness. So to check if mushrooms are conscious, we can just anesthetize them. What we have done, we took chloroform, and it's for a paper was published. We placed mushrooms or like substrate inoculated by fungi in closed container, insert electrodes in the substrate, started to record oscillations before stimulation. You see on this plot on the left, then put a piece of paper soaked with chloroform inside this continuum. Very quickly, after introduction of this volatile chloroform, oscillation stopped. Moreover, when we remove chloroform from the system, you can see it's off mark. Then oscillation return back. So basically, we can put mushroom asleep, and when we remove chloroform, mushroom wake up and continue the oscillation. And oscillation of electrical potential of the mushrooms might signify a consciousness or state of awareness of the mushrooms. That's basically all from me. And please ask me any questions if any, and I will stop sharing.
Thank you very much. And it was fascinating. It's really great what you showed us. I can see some hands. Please remember, if you want to ask questions, please use this in reactions. You can raise your hand. And first I can see Attila with his hand raised. Please, Attila, go ahead.
Okay, thank you. Hi, Andy. It was a very interesting talk. And I might have missed it, but my question is that sort of, are you making a sort of a story for the origin of the neural tissue? It's like, is it like an evolutionary story as well?
Partly yes, partly yes. Well, most of all, I'm trying to demonstrate that you don't have to be a brain to think, actually, because any system can spike, and therefore any system can think if there is some learning in the system. But also, indeed, evolution of kind of neural system, yeah, partly.
Okay, so then probably another silly question is that, so why is this spiking there? So what's the natural use of it? I get, well, I understand that you use it for doing logic and whatever, but what's the original purpose?
Original purpose probably to keep system integrated and to spread information between different parts of the system. For example, if you take plant, if you start breaking one of the leaves, we have action potential. Action potential propagates along vascular system, and then the whole plant become aware that one of the leaves was damaged, probably by insect. And then in this case, it allows the plant to develop immune response and release some chemorepellents, which kind of try to make insect go away. The same is mushrooms. If we damage mycelial network at one place, then fungi, well, not think, but anyway, kind of think that something going on, and they increase fructification, production of fruit bodies in other parts of the mycelium. So they respond kind of with reproduction to stimulation, yes. So integration, basically, just informing other parts of the organism about what's going on.
Okay, well, thank you. That's, again, great stuff.
Now, Gordana, please go ahead with the question.
Thank you very much, Andy, for a fascinating talk. I wonder, do you really need consciousness? Wouldn't be enough with saying that those organisms have cognition? Because if you think of Maturana and Varela and the idea that every single cell is cognitive system, then this cognitive system reacts to the environment and all those things. We had a talk on Wednesday by Michael Levin talking about basal cognition. So would it do with cognition?
Consciousness was just for fun, basically. I'm not really a fan of consciousness because there is no formal definition of consciousness. And like, well, so what I demonstrate about anesthetizing is the only actual definition people can produce about consciousness. And in this term, any living creature has consciousness because it can be anesthetized. And about cognition, basically, there is also no agreement about what is cognition, as I understand. There is no formal definition. Therefore, indeed, we can interpret anything as cognition. Because what I want to say is that everything is just matter of interpretation. For example, if you have stone rolling down the hill, that, well, from physics point of view, just following gravity, and that's all. But indeed, some person who doesn't know physics can say that stone is actually intelligent, and stone is actually calculating shortest path. towards downhill, avoiding like minor elevations. So yes, just to answer your question in short, cognition and consciousness is just matter of interpretation. But essentially, from my point of view, we have just physical laws in our life. And it's human beings who interpret everything. And living creatures just live their lives following laws of physics.
I would disagree. I think that there is a science of cognition, where you have, if you follow biologists like Michael Levin, you will say that's quite a well-defined term, and consciousness is much more diffuse and not so connected to natural sciences. So I would prefer, but that's my preference. I would prefer cognition.
Yes, I agree with you. I agree with you. Yeah. Well, personally, I don't use either of the terms in the papers, but yes, I agree with you. Cognition is more more practical, I would say. Yes, it's more pragmatic. Yes, I agree.
Thanks. Well, thank you, Gennaro.
Okay, hi Andy, nice to see you. Following the Gardona's question, when you put this question about are fungi consensus, or we can ask the same thing for physarum. So when you stimulate the physarum, is this a good stimulation for the organisms or negative for it? The organisms died, survive, or what happened? Reproduce?
Well, if we do make mechanical stimulation, it indeed survives because we don't put like heavy weights. So if we stimulate with nutrients, it's also good stimulation, and frequency of oscillations increasing. Yes, and yeah, so it basically survives. Yeah, we don't kill slime molds, plants, or fungi. We are actually, yes, very gentle with them.
Okay. My next question is, when you speak about mushroom language, what kind of structure follows this mushroom language?
What kind of what?
What kind of a structure?
At the moment, we don't know any structure. We just think there should be some language of mushrooms. And in that paper, we just interpreted mushrooms as like binary strings, like oscillations, spikes, binary strings, and just compared complexity of the binary strings of the spiking of fungi with complexity of the European languages. And at the moment, and language also were like pieces of text were converted to binary representation and compare it with binary representation of mushroom spiking activity at the moment. And with regard to structure, we are still trying to discover actually the structure of the language. More likely, it expressed in the length of the trains of the spikes and distance between the trains. But so far, we don't know exactly.
So you can recognize ambiguous reactions?
That would be fantastic. It's a dream to understand language of mushrooms, because mushrooms act as communicators between trees in the ecosystems and also between trees and insects and backward. They're like messengers. As soon as we understand their language, we will understand language of nature. And then we can live in total harmony with nature.
Okay, thank you so much.
Very nice comment from Andy. Right now, I think Hagia Sensei.
This is Masami Hagia. Nice to see you again. So I can't change my name into alphabet. Sorry, it's in Chinese characters. So I see spikes are everywhere. Spikes are very universal phenomenon. So my question is, what are the minimal conditions for generating spikes? Is it a kind of membrane or compartment, or so what are the general conditions or minimal requirements for generating?
And the system can stay in the resting state, then it can go away from the resting state, away from the equilibrium, and then with time it returns back to equilibrium. Well, just like, for example, if you take mechanical oscillator, the spring, when you like mechanically stimulate the spring, it will produce spikes. but it will be like damped oscillations. So with time, amplitude will be less and less and less. But like excitable medium, like Bizet reaction or living systems, because they have supply of energy, then when resting state is restored, there is enough energy, internal energy, to produce another spike, and then it continues. Yeah.
Thank you.
Okay. I don't see any hands, so maybe I'll ask my question. Maybe I missed it in your lecture, but I would like to ask about some form of memory, because you were talking mainly about operation of the system, but is this, for instance, spiking reflected in something which is preserved, which is conserved, which would be some form of memory? And if it happens, then next question, for instance, is there any genetical transmission that, for instance, if you induce some behavior in fungus or plant or any other organism this way, is it transmitted genetically later? Maybe you didn't do this, but I'm just curious.
Yes. Well, if you talk about physical or chemical systems, well, there is some kind of memory in terms that state of the physical system is changing. Like if you take Belousov-Zhabotinsky reaction, then frequency of spikes is changing, and even like their shape with time, because we have exhaustion of the catalyst in the system, and also formation of the air bubbles, and air bubbles cause irregularity in the wavefronts, which is reflecting the changing of the shapes of the spikes to more kind of randomized, basically. It's not real memory, just like development of the system. In terms of fungi and plants and slime mold, more likely there is some memory, but we have no idea because so far results on the spiking of these creatures is extremely limited. So we don't know at the moment, actually. More likely there is some memory. As you can see, you saw in experiments with loading of fungal block with kettlebell, 16 kilo. Every time we put kettlebell, then this fungal block reacts with less intensely. Therefore, there is some kind of memory. I put alarm for myself. Once it's like will be 7:45. Yeah. Yes. So we're still investigating this.
Thank you very much. Any other questions? I'm just looking for raised hands.
Yasuhiro Suzuki has a question.
Yasu?
Yeah, may I?
Yeah, sure.
Oh, thank you very much for very exciting presentation. Relating to the former several questions, I'm wondering, I saw many types of spike kind of languages and the nature. So do you think we can use such a spikes language to communicate with them? We can communicate with nature by using a spike language.
Yes, we can. As soon as we decode their language, to send them back some kind of sensible words, then indeed we can communicate. But right now we don't even understand their language.
Okay.
So we can actually send some kind of random probably trains of spikes and detect responses. But because sheer combinatorial kind of amount of possible combination is so high, it will take some time.
Okay, thank you very much.